let me very briefly. It's a 10 years work illustrating the efforts and initiative of 11 countries. US, not UK, France, Japan, the Netherlands, Sweden, on setting up a kind of the new innovative trend in life sciences, biology, and medicine. Biodesign-inspired, biotech-driven translational research and application, where chemical design, chemical engineering, and chemical analytics are considered to be a crucial element. So I have been requested to present you the global trend, and I'm just trying to do it. Okay. The new model of healthcare services globally, in contrast to Hippocrates' old model. Personalized and precision medicine being based on cellular, molecular, and now already quantum dots related biomarkers. As a new model of healthcare services is the unique integrated entity demonstrating an integration of fundamental clinical science, bioengineering, translation art, and IT armamentarium. Whilst illustrating application of the different tools on 3 strategic levels. Look, individual, population, and community related. And thus reshaping tomorrow's healthcare whilst doing it today is becoming crucial for biodesigners, biotechnologists, biomanufacturers, and for sure, consumers. To achieve the implementation of PPM concept into the daily practice, it's necessary to create a new biomarket-driven strategy based on biodesign-driven projecting. It's initiated by Stanford and Hong Kong universities. It's just the inner infrastructure of biodesign. Biomaterials being crucial element. Look, please, it's just the evolution of biomaterials market. Very briefly, unfortunately. And finally, biotechnologies integrating PPM as being the grand challenge to forecast, to predict, and to prevent, as you see, not to treat merely, is rooted in a big and a new science generated by the achievements of systems and synthetic biology. Systems, very briefly. Synthetic, integrative medicine, translational application to connect basic sciences and daily clinical practice. And developmental engineering paradigm. If you could read it, please do it yourself. whilst integrating crucial platforms of fundamental sciences, in American terms as basics, and innovative omics biotechnologies. Look, molecules and omics portfolio illustrating the latter. Through the algorithms of bioinformatics and artificial intelligence, AI, now much more machine learning, natural language, and even blockchain, which are being implemented finally as the newest entities into the daily medical practice to secure the biodesign-driven regular clinic and unusual subclinical, prognostic, predictive, and preventive manipulations of the next step generation being unknown for hypocrites. NH has included ppm associated with translation into a list of the 5 greatest priorities of development of biomedicine healthcare services in the current century, and have set up in the US 2 academical clinical research networks, including Centers of Personalized Precision Medicine, several examples. It's one of the best. Centers for Translational Medicine, it's the best in San Francisco. And Centers of Biodesign, dominating in Stanford University. To secure the lifestyle and healthcare services rearmed with the unique armamentarium. So, in daily life and practice, the crucial and indicative sign as litmus tests are becoming through biodesign-driven bioengineering, biopharma market, biotech market, biomanufacturing, bioindustry finally, and the design-driven innovative bioproducts finally, which we have as the consumers. Please look at the global market for bioproducts in 20th, 21st. Unfortunately, I'm having no 22nd, 23rd. And innovation is thus considered to become the key to address the unmet clinical needs in this STEM: biodesign, bioengineering, and biomanufacturing can be explained as an approach that includes various research and design-driven applications related to taking inspiration from natural biofunction, biosystems, biocomponents, or bioprocess in solving the problem. Just for example, biomanufacturing, it's a special course of lecture 184 hours at MIT. It's phenomenal. 11 professors from different universities in the world doing it at MIT in terms of manufacturing. In this sense, biomaterials are used for the repair, replacement, and stimulation of biosystems. Biomaterials have revolutionized the areas of biodesign-inspired bioengineering. for the development of novel strategies to combat life-threatening disorders and to secure healthcare provision whilst are being used for the treatment of different diseases. Biomaterials are either found in nature, I mean natural biomaterials, or can be created by organisms, tissue-derived or biological ones. Types of biomaterials, please look very shortly. Or can be made in the lab from recombinations of starter materials found in nature. The recent increased interest in biomaterial innovation is rooted in working with the frame of biosystems through both natural metabolisms and created ones. It's just the illustration how it's being doing in the frame of systems of making biomaterials. Let me concern microfluidic technologies in particular. The pioneering is just MIT, with emphasis on transdisciplinary application in the fields of chemical biopharmacy, while stressing the recent applications and development on genetic analysis, cell manipulation, biosensoring, pathogen detection system, diagnostic devices, high throughput screening, and biomaterial synthesis for bioengineering, and for sure, biomanufacturing. Look, please. Lab on a chip LOC as a product of the latter is a kind of microfluidic devices which are broadly used in life sciences whilst integrating one or several lab functions, multi-lab functions, on a single chip, which is now fabricated on LOC's platform using biopolymers as a kind of biomaterials. Those platforms aimed for the detection of mutation, doesn't matter which one, microbial or human, has been developed and can help in the identification of human pathogens, oncogenes, screening genetic disorders, and confirming thus cancer diagnosis. Moreover, recent research in this field has developed and utilized a variety of different polymerization synthesis methods tailored to the application of molecular inherited polymers, MIPs. And I would stress, develop, and assess the principles of molecular imprinting, MI, for disease diagnostic, including recent progress in integrated MMP sensor technologies for high affinity bioanalytic detection in complex biofluids. Consequently, in coming years, next generation biotechnologies will reorient medical practice more towards disease prediction and prevention. You understand how valuable is the latter approaches rather than curing them at later stages of their development and progression before the death, even at wider population levels for general public healthcare system. In this sense, let me stress that with the advances in omics technologies, very briefly, quantitative proteomics is emerging as a powerful technology complementing the first one, genomics. It's just very briefly overview. Quantitative proteomics opens a wide range of application in clinical diagnostic treatment, stratification, and the most valuable, drug discovery of drugs of the next step generation, smart or intelligent drugs. The candidate proteins might provide novel, principally novel targets for new targeted therapeutic agents or aid the development of assays for disorders, biomarkers, and identification of potential biomarker target ligand drug composites. Just the illustration schematic structure of receptor targeting drug conjugates, carrier, linker, and potentially therapeutic payload. Functional proteomics allows the design of strategies to target multiple pathways with a combination of pathway-specific drugs. I would stress pathway-specific, not molecular-specific, pathway-specific. So it's interactomic-specific, which might increase chances of success and reduce the occurrence of drug resistance. It's just the illustration, of the intrinsic kinds of proteomics. Meanwhile, chemical proteomics as a part of the global proteomics, by analyzing the drug interactome, strongly contributes to accelerate the process of new, I would stress the new term, druggable, targets discovery and drug delivery. Chemical proteomics, by analyzing the drug interactome, strongly contributes to accelerate the process of new druggable targets discovery. Please, like the illustration of what I have just mentioned, it's the crucial element to confirm the market. Fortunately, targeted drug delivery drugs are oriented to attack only specific targets, can improve the enrichment of drugs at the target site and reduce toxicity of normal tissues and cells. The targeting component is called a chemical ligand. Aptamers have high target affinity and specificity, which identify as attractive and promising ligands. For example, it's interesting for me unexpectedly, since I'm not a chemist, chemical ligand and its application, 86 hours special course at Stanford University. Crazy. Therefore, aptamers have potential application in the development of, as I have already mentioned, smart, intelligent targeting system. Look, please. The cell, cancer cell. It's the cancer cell specific receptors. As the first step, ligands, chemical ligands. Finally, targeted drugs. Targeting, the first step. Targeted drug delivery system is the second step, one of the second crucial elements for chemical engineering. Potential nanocarriers being used for delivering. Meanwhile, aptamers, including DNA, RNA, and peptide aptamers, are a group of promising recognition units that can specifically bind to target molecules and cell. The specific binding ability of aptamers to cancer-related markers, I mean biomarkers, and cancer cells ensure the high performance for the pre-early. In professional community, it means subclinical. I hope you understand me. Subclinical, not clinical, where practitioners are used their potential. Subclinical, where no clinical manifestations. Meanwhile, the efficient targeting ability of aptamers to cancer cells and tissues provided a promising way to deliver imaging agents and drugs for cancer imaging and therapy, while stimulating the development of the latest technology, phenomenal technology, molecular imaging. Please, it's just the illustration of the technical scheme. It's the clinical illustration of the application value. Unfortunately, at this point, only in the area, restricted by the area of clinical oncology, unfortunately. This is the market of molecular imaging. Look. To integrate diagnostic potential and treatment potential within the frame of one single drug. Companion diagnostic and theranostics. This is the market illustrating the latter. It's phenomenal achievements. The first companion diagnostic, 2013. A footstone of stimuli-responsive nanocarriers, endogenous, exogenous, responsive, smart materials possess many properties, such as responding ability to specific triggers, controlled drug release, long blood circulation, increased tumor accumulation. So a combination of omics-related biomarkers are becoming of great significance to be translated into the daily clinical practice to predict risks of the chronification and thus of disabling. The applicability of canonical metabolic engineering strategies. Let me have 10 seconds rest. Please read yourself the bottom. This has extended with a combination of biodesign-driven systems biology and synthetic biology simultaneously in a systemic and versatile manner, and are collectively termed as systems metabolic engineering. Metabolic, I mean pathway-driven engineering. Look, it's just the illustration in brief of the latter. Accordingly, recent advances in biotech have paved the way for enabling the production of an increasing number of monomers and polymers by providing several tools and strategies associated with systems metabolic engineering to develop and even, finally, manufacture metabolic disorder therapeutics, which are now under FDA clinical trials control. Yet, please look at the market of the latter. Dynamics. As a result, very briefly, modern therapeutic modes and approaches, principally new, cellular therapy, metabolic disorder therapeutics, phenomenal achievements, in particular in type 1D. It's so much interesting. At present, no ways to treat type 1D. The only way to maintain type 1D patient by insulin. Professor Dick Anderson is a genius personality, head of, don't keep in mind, MIT. He is not a medical doctor. He has developed a technology to prepare stem cell being translated, specially prepared stem cell being translated into the beta zone of pancreatic, where in 24 hours, they would generate insulin. Preclinical studies, 400 monkeys with experimental diabetes. Full-scale restoration in a couple of days. No insulin more, but 3 companies, Novo Nordisk, Eli Lilly, and don't keep in mind the third one, in Europe, who are generating manufacturing insulin, have pre-agreed with the US Congress not to allow the clinical trials. No, I'm sure that him would win it, but not now. As an illustration of drastic changes and new trends in biodesign, biopharma, and biotech, you may see below. Just look through it. It's a kind of innovation maps in the area. The growth of profiled publications, so cosmic bursts. Market. We are experiencing, as you understand, a renaissance primarily driven by biodesign, biotechnologies, and biomanufacturing. And the grand change and challenge to secure our health and wellness are rooted not in medicine and not even in science. Just imagine where? In high-tech culture. Thank you so much for your patience.